you for coming to my capstone presentation. My name is Jessie Nelson, and I'm a junior here at Wolcott High School. My capstone project was an internship at Brown University, where I studied biomedical and biotechnology for engineering. So the typical capstone process is first a proposal, second is followed by your research, third a product, and fourth the presentation, which is today. However, my process was a little bit out of order. I first had my proposal. Obviously, I needed an initiative plan of action, which followed by my product. That's when I started my internship at Brown, and that was around October, which then triggered my research paper, and then today, the presentation. So at first, I was struggling with what I wanted to do. I have a lot of different interests. I didn't know if I wanted to do something revolving around sports or a fundraiser for a particular organization, but then I decided to do it on a potential career option, which is biomedical engineering. For those of you who don't know what biomedical engineering is, it's a relatively new field, and it combines the fundamentals of biology and technology to make medical advancements. Some examples of what biomedical engineers do are working with 3D printers to use, instead of regular ink, obviously, they use cells and different body fluids to create different organs or tissues. Also, they work on modifying prosthetics to make them work like different limbs would naturally work. So that's just a scope on what biomedical engineering projects are doing today. Some challenges I faced working with this project was I had to find a facility that would allow me to have an internship there. First, I contacted local offices, but due to legal things and HIPAA laws, I wasn't able to internship there, and so I got negative feedback there. I reached out to a couple colleges in state, like UConn, but again, I wasn't getting the response I was hoping to. And then I finally made a connection with Robert Cretan, who is the director of imaging in the biotechnology department at Brown, and then later furthered that connection because Mr. Pelosi, the foreign language teacher here, his daughter teaches at Brown, Diana Pelosi. She's a professor in anatomy there. So she took me under her wing and connected me with her colleagues. So this is Sydney Frank Hall, and this is the biomedical and biotechnology department at Brown, and this is where I completed my internship and did my research. At Brown University, I worked with Diana Pelosi, Robert Cretan, the director of imaging, Emily Reiger, who was an undergraduate student, and lastly, Hillary Nicholson, who's a graduate student. In October, I started my internship with Emily Reiger, and she's doing research revolving around zebrafish. So this is a specimen used in biomedical engineering, and what she was doing was using red fluorescent protein to tag the calcium protein in the zebrafish body embryo, and then she exposed these embryos to the drug dioxin to see the effects that this drug had on this particular calcium protein within the body. So this is just a picture of Emily retrieving the collection trays of the embryos in the zebrafish tanks. This is Emily pipetting the right amount of the drug she wanted to then micro-inject the embryos with. And so as you can see, the biomedical engineering field has very precise research scopes because it was one drug focusing on how one protein is affected in the body. This then triggered my research paper, and I focused it on why biomedical engineering uses zebrafish as the specimen. And I found that zebrafish are used because they're very easy to maintain and they reproduce rapidly, so their embryos are always available for research purposes. Also, the embryo is transparent, so it's see-through, and you can look at the processes of development from the outside looking in. And lastly, they develop over 24 hours, so the students actually take shifts and make observations in 6-hour shifts of everything that's going on. Another thing that zebrafish offer is they have regenerative processes that humans do not have, or they do but we don't know how to trigger them yet. For example, zebrafish can regenerate retinas, they can regenerate fins and different valves in the hearts when they break or malfunction. So we're looking at They try to see what enzymes or what catalysts that zebrafish have and what capability they have that maybe humans do too, so that we can do that as well. Next, I shadowed with Hillary Nicholson, who's a graduate student doing her study there for her PhD. And the first thing we did was a Western blot. So she works with cancer cells, and the first thing that we were doing was looking at the BID protein in cancer cells. And the BID protein is a protein that breaks when the cancer cell dies. So a quick diagram that she drew for me was, these are the cancer cells, and these are the BID proteins breaking and the cells dying. So what we had to do was we tagged this BID protein with an antibody, a fluorescent one again, and then we put them in a cassette, like shown here, and these are the different colors. The red is the BID protein. And we then went to a imaging room, and it's almost like an x-ray. You put the cassette in, and it produces an x-ray image. And this is where she can measure the different lengths of where the protein is breaking, and then she then uses that in different mathematical equations for analysis and different graphs, and she makes sense of that. Another thing that we did with the cancer cells while we were there is we were doing a project of seeing different concentrations of different drugs and how they affected the life or death of the mitochondria and of the cell in general. So we know that the mitochondria is the powerhouse of the cell. It produces ATP, which is where the energy is. So if the mitochondria is working and producing energy, the cell is alive. So we used a color indicator in the mitochondria. So if the mitochondria was purple, that meant the cell was alive, and if it was yellow, that means the drug and the percentage caused the mitochondria and cell altogether to die. So we tested out different percentages of different drugs and see how they affected those mitochondria and different cells. So this is Hillary adding the different drugs with a multi-pipetter, and then this is a imaging, again, machine that takes the tray of cells and you put it in, and it measures the different colors, because it wasn't just light purple and dark purple or like yellow, and like there were so many different shades that this took it and analyzed it based on the true colors that it had, and so it then formulates more data that she can use in her research. So that was about January that I finished my internship at Brown, and this was about the time that everybody else started working on their projects, so I wanted to do something else. And we came to the conclusion that I would present to the Project Explorer students at the middle school. And in doing so, I had to plan a lesson for them and tell them about how my experience at Brown was so beneficial. So I showed them a PowerPoint similar to the one today, just with more pictures and more in depth. Also, I had to focus on explaining the content at a level with their background in biology and different sciences so they could make sense of it. And lastly, their attention span. We all know that we don't like sitting there for like an hour listening to somebody, so I definitely wanted to include a hands-on activity that related to my topic and also got them involved. So this was me showing the PowerPoint and explaining the Western blot to the kids. Next, I demonstrated, we did a strawberry DNA extraction lab. Now most of people, when they think DNA, they think fingerprints, hair. They don't really think of fruit to be something that contains DNA, but yes, all living things have DNA. So what we did was, in the front of the room, I demonstrated, and we took 2 or 3 strawberries and smushed them up in a bag. We added a DNA extraction solution, which was soap, salt, and water, and this ate through the cell membrane and allowed the DNA from the strawberries to flood out of the cell. We then set up a filtration system, and that allowed the DNA in the solution to stay at the bottom and the clumps and chunk at the top, and we discarded of that. And then we put that into a test tube and added cold rubbing alcohol, which caused the DNA to precipitate to the top, and the students were actually able to see the visible stringy white DNA at the top. So this is me adding the rubbing alcohol and then being able to see the stringy DNA at the top. This is Mrs. Penta. I knew she wasn't going to be happy with this picture, but I wanted to thank her for being one of my core mentors throughout this whole process. Overall, from my experience, I think it was definitely beneficial, and I encourage each one of the sophomores in this room to take this as a senior. It gave me hands-on experience of what the field is like, sitting in a lab, doing research in a college setting. It gave you like a real perspective on that. Also, real-world skills. I had to reach out to all of the colleges myself. I had to get rejected by the colleges myself. It wasn't an adult figure doing that and helping me along that way. So it gave you the communication skills that you need. And then also public speaking. Like today we need to be able to convey our ideas and topics. So this gave you a chance. I have to present today. I had to do presentations throughout my whole year. So it definitely improved my public speaking skills a lot. So I encourage anybody to take this class. A special thanks to Mr. McIntyre and Mrs. Thies for guiding me along the entire way. Also Diana Pelosi in the Biomedical Engineer Department at Brown University. Mrs. Penta, Madison Hanlon for coming and taking my pictures, and my supportive parents, and my mom for driving me up to Brown every time I had to go. At this time, I'll take any questions or comments.
Do you see yourself going to Brown for like school? Are you interested in going there?
Yeah, Brown is definitely a dream and a reach school, and I would be fortunate to go there and be lucky enough, but it's definitely a dream school and something that I'm reaching for.
Did you have background in any of this before you started?
Not really. The regular biology classes at the school. I then furthered myself before I went up to Brown and researched some things, and then after I left Brown, they explained it to me, and while I was there I had to double check, she'd explain, Hillary would explain something to me and I'd tell it back to her in my own words to make sure that I understood what was going on. So I really didn't have any